using his creativity in the click chemistry molecular imaging tracer to support oncology and neuroscience drug development. Welcome, Dr. Kolb.
I'd like to thank the organizers and also for the kind introduction. So I'd like to talk about click chemistry and how we've used it to develop, to be useful, to make something that's useful. But before doing so, I'd like to just back up 26 years roughly and discuss how we actually came up with click chemistry. Because as we go through our careers, there's every day is almost the same, right? But there are singular moments when things change dramatically. Right? And for me, it was a board meeting in a small company that had only one employee, that was me, with Barry Sharpless in New York City in 1997, August. The company was founded by Barry Sharpless. He was then, he had no Nobel Prizes yet, but he was famous for his asymmetric catalysis already. And the company was founded by him to develop new catalysts that could be useful. And at that board meeting, he decided he didn't want to do it anymore, and he was done with it. And he wanted to instead invest the resources of the company to develop new chemicals, new chemistries that are reliable. Right? And that moment was the birth of click chemistry. Right? It was on August 18th, 1997. And the idea of click chemistry is to do the opposite of what many chemists do. Many chemists get lost in the beauty of complexity. Right? They make complex molecules, ivermectin, azadirachtin, all these big things. And click chemistry is the opposite. Right? The idea is here to keep things simple, right, and use the speed that simplicity gives you to quickly explore the universe of chemicals and reactivity and properties. Right? And that was the idea. And so we focus ourselves single-mindedly on just the best reactions. Right? And these were, nature gave us those ideas. Right? Nature told us what those were. They were carbon-hydrogen bond-forming reactions. Right? Basically making carbon-oxygen, carbon-sulfur, and other bonds. So then we wrote a publication that we called the Click Chemistry Manifesto, right? And there was a paper that describes exactly how we were thinking, how we would make diverse chemical function and properties from a very few good reactions, right? And so that was written, published in 2001, right? And this looked all beautiful, right? I said a lot more hair, he did too, but what years before, it took us 3 years to write this paper, right? So here's an earlier draft that he and I wrote. So you can already see the carbon-hydrogen bonds being formed and so on, but it was a lot of work to get this done, and a lot of editing by Sharpless. It was a nightmare, but we got it. You know how this works, right? And we got it done, but already then, we already were focusing on azides and triazoles, right? Because forming a triazole from azides and alkynes or alkenes, we already realized then this was going to be an important reaction. This was years before the famous QUAC reaction, the copper catalyzed click chemistry reaction was invented, right? So this was, in a way, we foresaw the future in a way, but we didn't know such good reactions could exist, right? And so then eventually, in the labs of Morten Meldal and Sharpless and Fokin by Luke Green, they found that if you mix an alkyne with an azide and copper, a little bit of copper, you get a triazole within a few hours, right? And this is, I think, the key reaction that made click chemistry what it is today, right? And so now we have a reaction where we can simply, like a glue, combine things and make new compounds. So Sharpless himself, he calls this a magic reaction. So these are his words. It's actually a slide from his Nobel lecture last year. So he calls it a magic reaction that produces a triazole anywhere and any time, right? And it'll never fail you, right? And that's why this is so powerful. It has been used for bioconjugation reactions and for making ligands for drug discovery and so forth. But the other reaction that we worked on was in situ click chemistry. Here we used an enzyme or a protein as a molecular scale reaction vessel, right? So we used basically we just threw in a bunch of azides and alkynes into a mixture with a protein of interest, for example, acetylcholine esterase or carbonic anhydrase, whatever we targeted. And then the protein would assemble its own inhibitors inside its binding pockets, right? And we call this in situ click chemistry. So this allows us then to make high affinity ligands because this has a bidentate affinity to these proteins. And so we were able then to make high affinity ligands that we can use for drug discovery or tracer discovery. So we published a number of papers on that. And this then led my work for the next, I would say, 20 years, right, time goes by quickly. And so we use this in situ click chemistry and click chemistry to make tracers for molecular imaging. Right? And the way molecular imaging works is that and that's why I got excited when I first saw this, that we do nuclear physics in the morning, right? We make our own isotopes, right, in a cyclotron. And then we do radiochemistry to incorporate them into a ligand. The ligand is discovered using click chemistry, right? And then we do, of course, in vivo imaging and positron electron annihilation, so antimatter matter annihilation to generate gamma rays that we can measure and then reconstruct to get the image, right? So to me, scientifically, this is incredibly exciting, right? And it's useful, as we saw earlier in the talk, right, where we can do amyloid imaging, tau imaging, and so forth, right? And so that's what we did. And so click or imaging can be used then for basically measuring the expression of targets anywhere in the body, for example, amyloid in the brain or a cancer target anywhere in the body, right? For example, HER2 in the body. It can be used to do in vivo competition studies, so you can measure target engagement in the brain or anywhere. You can also look at downstream changes. So that's if you hit the target with a drug, what happens, right? And we can measure that in vivo. We can look at mitochondrial metabolism, right? And that's I've done this with Yvonne, who's here. And there's many applications that we have with imaging beyond just measuring the presence or absence of a target. And one application that that my team and I developed very early on was a combination of in situ click chemistry and quark to develop a radio tracer for carbonic anhydrase, which is important for hypoxia measurement and so on. So we used the enzyme carbonic anhydrase to template the formation of its own inhibitor using in situ click chemistry shown in green, and then we made the same compound with F18 using quark, right, using copper, right, and then that was then injected in the mouse in this case, and we were able to image carbonic anhydrase in a mouse, and later also in humans. And this gave rise to a nice paper in MedChem at the time. So that's one application. Then of course many more. So I will show you a few applications. Switching gears, so I'm going from click chemistry to imaging to precision medicine, and in precision medicine, the goal is really it's the patient's benefit, right? I think the key goal is to give a patient a drug that actually works, right? And that's, in a way, a paradigm shift from the past where people were treated based on the clinical presentation of the disease, right? For example, treatment-resistant depression was treated with SSRIs, right? No matter what the underlying biology was, or breast cancer was treated with various regimens that were prescribed. With precision medicine, what you do is you look into the underlying pathology, right, biology and genetics, and then subgroup patients and match those patients then to the right treatment. And that worked very well in cancer. What we know about, of course, Herceptin and HER2/neu, and also a little later, the approval of Keytruda, pembrolizumab, was based on the biomarkers and not on the clinical indication, right? And this was a paradigm shift, right? Because in the past, drugs were approved based on the clinical presentation. Here we're looking at biomarkers. The beauty of using biomarkers in precision medicine for drug development is that you then basically get those subgroups that are clean. And then Dr. Feldman already showed us earlier that if you have a drug trial that has 20% of people that don't have the target present, then you're diluting your population by those 20%. Right? So you want to avoid that. In precision medicine allows you to do that. But we then also, of course, are more likely to make drugs that actually work, that help the patients. And that's what we focused on then at Janssen over the last 9 years. And so my team focused on 3 key questions. Right? And one first question is, who's the patient? So how do we find the patient that matches a certain treatment hypothesis, right? And for example, amyloid or whatever. Then the second question we have is, how do we measure that the drug actually works? Does it hit the target, right? And how do we measure whether it's toxic or not, right? And so these are things that we look at a second question. And the third question is, how do we measure response, right? So does the drug do what it's supposed to do with the patient? So, and my team, in collaboration with many others, we look at new assays for patient screening, for diagnosis, for following people, right, to see whether the treatment responds, for toxicity, right, but also for scaling it up to large scale, to global scale, right? Because if we have an assay that allows us to find a patient that would benefit from a certain treatment, we want to make sure that that assay, the test, is available globally, right? And that's what we're doing right now with a blood assay for Alzheimer's disease that allows us then to measure in blood whether a patient has the Alzheimer's pathology in the brain, right? So we're currently scaling this assay so it can be run anywhere on the globe. And so that's, and I'll show you some examples later today. So I have more minutes. And the first question is, who's the patient, right? And here, this was near and dear to me because my late wife, she had breast cancer and it was metastatic. So we scanned her and other patients to see whether any of the metastatic tumors had HER2 expressing, right? And she was HER2 negative, right? And turns out that of the 9 patients that we scanned, 3 patients had mets that actually were HER2 positive. So they went on Herceptin treatment, right? And that shows you that imaging, whole body imaging, can really be used to find new treatments for patients that previously weren't eligible for this treatment, right? And so that actually helped 3 people here, right? 3 patients with breast cancer. And so that was the work we did at Memorial St. Kettering and with Gary Hulana as the PI. That shows you how imaging and diagnostics can help you find the patient and match the patient to treatment, right? And so that's, I think, a very good example. The other thing is, does the drug hit the target? That's especially important for the brain because you can't really take brain samples easily. So in other words, we just do PET imaging to find out whether the drug that we're developing, in this case, AMBA-TAP gamma-8, hits the target in the brain, right? And we just do it in a way in vivo titration, right? Where we dose the drug and dose the tracer and then measure the PET signal in the brain. And you can see on the right here, see how we see a increasing target occupancy with increasing dose of the drug, right? And so we can take away the guesswork from drug development. So now we know what dose do we need to get a certain target occupancy, right? And so that's the other thing. The third important application that we worry about is how do we measure response? How do we track a patient, how he or she is doing? So one key thing we need to do is we need to minimize retest variability, right? Because as you do a test in the same patient 2 times, you oftentimes get different results, right? And for example, if you look at the mini-mental state exam score in a patient, you do this patient once and then again, you get often different results by as much as 4 points. That's about 13% of the scale. In other words, the noise of the test-retest in using a clinical score, such as the often used MMSE, is 13%. But so you have to go above that in your treatment effect to see something that is actually meaningful. But with imaging, that's much less, right? Because there the test-retest is 5% variability. So in other words, you get a much better signal, right? And that's a much more reliable signal. Of course, no patient cares about the image, right? People care what happens to them, but the clinical score really matters. That's what matters to the patient. But imaging can be used as a surrogate then, right, to get to the clinical score. And because it's much more reliable, less noisy, right? And that's really what was used in the approval for aducanumab, right? And lecanemab, as we just heard, for aducanumab, the approval was based partially, at least, on the fact that imaging showed removal of amyloid, which then was reasonably likely to have a benefit on the road for the patient, right? And so in a way, it was a reasonably likely surrogate endpoint that the FDA decided to use. Rightfully so, I think it was a good decision. And so that's so very exciting stuff. The other thing we worked on over the years using click chemistry, basically we imaged many of the hallmarks of cancer, right? And so we looked into signaling, growth factor-based signaling. I mentioned already HER2. We looked into ways to hypoxia, which also is very much linked to response to treatment. We looked into invasion and metastases, right? MMP imaging, carbonic anhydrase already mentioned, PSMA, proliferation, in angiogenesis, and as well as cell death, apoptosis, right? So we made image tracers for all of these events, so we could really capture all these hallmarks of cancer using imaging, and of course biopsy. So here's some examples. The blue shows the click chemistry linkage, right? It's this quark linker, and you can see how click chemistry really enabled this work over the years, right? You can see here on the left, hypoxia tracer that we developed, and we imaged cancer patients with those tracers. In the middle, we see an angiogenesis image using this RGD K5 labeled compound, again, using click chemistry. And then on the right, we developed a caspase 3 substrate, actually, that images apoptosis. Everything with click chemistry, right? And so that shows the power of click chemistry for discovery, right? And it didn't take us very long to do this. For example, for the hypoxia, I had the idea for this epoxy agent in March many years ago, and in October we already had the first human image. Right? So it allowed us to go this quickly. Right? Well, it may have been November, but it was in the same year we had the first human image. Right? And that shows you how quickly this would never happen in drug discovery. Right? So this shows you how quickly we can move with good chemistry. And the last thing is then Alzheimer's disease. So I won't go into all the details with ATN. You just heard, right? A beautiful lecture. So we don't need to. I don't need to explain how this works. The good thing is that the efforts that we just heard about, ADNI and ABLE and all the HABs and so on, they gave us really a good idea of what the timeline is for these pathology, for these hallmarks, right? And we heard about amyloid, tau, and neurodegeneration, and we now know from all these efforts that amyloid can show up years before you have any clinical symptoms, right? Like 10, 15, even 20 years. And so your brain can be full of amyloid, yet you have no symptoms, which is good, right? At least gives you some time. On the other hand, once you have tau, a neurodegeneration, you will have symptoms. And the tau pathology is closely linked to the severity of symptoms, right? So in other words, with tau, you can really measure more accurately how you're doing, right? And so that's what we are using now at Janssen, and of course others are using as well. Part of this, the tau, is based on the work that Heiko Brack did in Germany with his wife, right? And they did like autopsies on like 1,000 people and came up with the Brack staging system that links the tau pathology, the tangled pathology in the brain to the severity of the disease, right? And so Brack stage 1 and 2 is clinically silent. You have no symptoms. If you are Brack stage 5 or 6, your brain is no longer functioning, right? So then you are in full dementia. And what this looks like in real life is this. These are, of course, autopsy samples. You can see how the silver staining that they did, how the brain is impacted by the tau pathology, as shown by the staining. So these postmortem results clearly show that tau pathology is directly linked to the severity of dementia. Of course, this is too late, right? This is autopsy, so people are dead. You can't help them anymore. So we needed something at that time that would do the same thing in vivo, in living people, right? And so my team and I developed the first tau imaging agent that we just heard about, T807, which is now sold as fluortaucipir by Lilly. This imaging agent was developed at Siemens by my team, and using click chemistry as the beginning. And then the team at Lilly developed it to get it approved in about 3 years ago now. And now it's also reimbursed, right? So this agent is now available commercially and also has been used in ADNI, right? And we just heard the results. Those 4 images are the first ever images that we collected with this agent that we developed, right? So FTP. And I actually personally analyzed those images because I had no one else to do it, right? So I went and did this. And when my daughter, she was worried about that her brain might look like, she was still very little, she was worried that her brain might look like the one on the right. And I told her this wasn't going to be the case. So this shows you very beautifully how imaging can give you the Brack staging in living people. Right? On the left, you see a healthy person like us here, and then on the right, you see a person with a Brack stage 6. Right? The brain is fully loaded with amyloid, with tau in this case. And then here we had Mark Minton and I, we did same patient amyloid tau scanning. So you can see how amyloid shows up at the bottom much earlier than tau, right? And that really is really the basis for the Clifford-Jack plot that you see on the right, right? So where the timelines. And so this is the science behind those beautiful graphs. And we also know now, this is work by Chris Rowe and many others, that amyloid is a prognostic marker for decline. So if you have amyloid in the brain, it doesn't track decline, but it will predict that you will decline, right? Years down the road. Right? So the more amyloid you have, the more you will decline, more likely you will decline over time. Tau, on the other hand, is correlated with decline, right? So there's a little bit messy here, but there's a linear correlation between the tau PET signal, SUVR on the y-axis, and the MMSE. MMSE, as you remember, is quite noisy, so the correlation isn't that great, but you can see that the higher the PET signal is, the worse your MMSE score is, right? And the good thing also is, at least for the science, that a larger tau PET signal predicts a faster decline, right? In other words, tau PET is not only a marker of a monitoring marker, but also a prognostic marker, right? So it gives you a lot of information. And this was shown in thousands of scans. I don't have time to go into that, but in ADNI, 1,400 scans were analyzed. And this was also shown that the more tau you have, the faster you will decline. But this doesn't really help because it's great to have. But if you want to help someone, you want to intercept the disease before there are clinical symptoms, right? And so we started many years ago a trial where we were looking for people with pre-symptomatic AD. So people that had amyloid and tau in the brain, a little tau in the brain, but didn't have any clinical symptoms yet, right? Because then we would treat those and try to prevent further progression. The question is, how do you find those people, right? Because in this case, this was done about now 6-7 years ago. That was before plasma assays. We actually took 1,000 people and took the CSF and analyzed CSF for amyloid, as we saw earlier, and tau, and found that only 4% of these cognitively normal people had amyloid pathology and tau pathology, right? So A plus T plus, but no N. And so in other words, you're looking for a needle in the haystack, right? And that's really, really hard to do. And it was very hard to do. So that's why we needed a non-invasive and scalable blood test for this, right? Because if you had that, we would just take a blood sample and we would know what's going on, right? And that's what my team developed, right? So we developed a blood test for Alzheimer's disease based on a phospho-tau fragment. It's a tau fragment that's phosphorylated on tyrosine 217, right? And we're able to pick this up in blood. The amount of this tau from the brain in the blood is tiny, but we only have femtograms per milliliter in the blood, femtograms per milliliter. So you need a very sensitive methods to pick this up. And so we're using the Quanterix single molecule analyzer CMOA machine to pick this up in our own in-house developed antibodies. Right, so with this assay, we are now able to quantitate P217 tau in blood. And that we have now shown, thanks to a collaboration with Chris Rowe in Australia using the ABLE samples, about 400 samples, that this biomarker is associated with amyloid pathology as well as with tau pathology. Right? So it predicts amyloid pathology and tau pathology. Right? So we're using it now as a screening marker. Right? So we take blood, we screen a patient based on this assay. If the assay is positive, then this person goes on to tau PET to confirm, right? And that takes away the pain, but it makes it much easier for the patient and for us to find the patient that will have the AD pathology. Once we have them, we scan them, right? And here's a tau PET scan, a longitudinal scan. So the blue is at baseline, and then red, you see the new spread over 16 months. In other words, if you have tau at baseline, you will go down quite quickly, right? And we can measure that using tau PET. We can quantitate it, shown here in red. And we can also predict it. We know that the spreading happens along the neural connectivities, along the neural networks. And so we can now do an individualized analysis of the spreading of tau in people. And that will help us with the drug development. So we can try to block that, right, and measure the blocking. So this is, I think, the end of my talk. My time is almost up. So my team has developed over the last few years is a PET agent for tau that allows us then to quantitate tau pathology, drug staging in vivo, and track it. We also have developed a plasma assay for P217 that predicts the presence of amyloid and tau pathology in people. And we're using that now to run our clinical trials. But we have anti-Alzheimer's clinical trials running at Janssen that uses these assays and imaging agents in their trials. So I will skip this because it's FDA stuff. So basically, in AD, we have many of the applications for biomarkers covered, right? And this is actually, the FDA has a biomarker qualification program where they define the use cases for these various biomarkers, right? And so the A and T are diagnostic, they're also prognostic, and they can also allow us to monitor, right? I showed you tau PET. And amyloid PET has been shown or approved or allowed by the FDA to be a reasonably likely surrogate endpoint. That's how aducanumab got approved. And tau will probably follow down the road as well. So in summary, I think there's a good future ahead of us in neuroscience, right? And we saw, of course, the talk by Dr. Feldman already earlier. And so in AD, the knowledge about the pathology in AD allows us to really put science behind development, right? To find patients, track them, and help them. And this will also happen in psychiatry, because there we also have leads of biomarkers that are linked to symptoms. In schizophrenia, for example, it's complement in inflammatory markers, synaptic markers, and in depression as well. And so there are going to be a lot more precision medicine tools and trials in neuroscience. There will also be more efficient ways to get accelerated approval. We saw aducanumab with using amyloid PET. The same thing will happen in all likelihood with tau PET. And drug development will shift to more and more early intervention, as I mentioned earlier with our trials. And I think importantly, we have to work hard to make those tests available globally so that people can benefit from them. And that's really the summary of what we are trying to do as Janssen. So here I'd like to thank my team. They are one of the drone videos from Delmar Beach, right? So you can see they're all happy. And then a picture from Stockholm a few weeks ago. It was with Barry Sharpless. And then the teams that work on this. Thank you.
Thank